Hi, welcome to the session on the modality of cancer stem cells. My name is Dr. Ellen Feigal. I'm Senior Vice President of Research and Development at the California Institute for Regenerative Medicine. I'm very pleased with the distinguished panel of speakers we have here today. I think we're going to have a very exciting set of presentations and hopefully a very interactive set of discussions. Without further ado, what I'd like to do is give a brief background for each of the speakers, and then I do have just a list of some potential questions to consider during the discussion period. Each speaker is going to take about 15 minutes, and then we should have time for questions at the end to have a panel discussion. The first speaker is going to be Dr. Catriona Jamieson. She has a broad background in molecular stem cell biology and immunology, and in her laboratory she analyzes events evolved in the initiation and in the progression of blood cancers. She's co-PI on a CIRM Cancer Stem Cell Disease Team grant that's focused on leukemias. She's also, in her background, did her postdoc fellowship in the laboratory of Dr. Irv Weissman, and we're very pleased to have her today. She's at the UCSD Moore Cancer Center as well as at the Sanford-Burnham. The next speaker is going to be Dr. Robert Wechsler-Reya at the Sanford-Burnham Medical Research Institute. He's professor and director of the Tumor Development Program. His work really focuses on the signals that control growth and differentiation in the cerebellum and how these signals are altered in the pediatric brain tumor called Medulloblastoma. His work has received several awards. He's received an award, he's been a Kimmel Scholar Award recipient. He's received an award for excellence in pediatrics research from the Society for Neuro-Oncology, and he's also been awarded the Leadership Award from CIRM. He did his undergraduate work at Harvard. He got his PhD from the University of Pennsylvania in immunology, and he's done postdocs both at the Wistar Institute as well as at Stanford University. The third speaker is Dr. Daniel Nolan. He's Director of Research at Angiocrine Bioscience. He's an alumnus of Stony Brook University, and he did graduate studies there in vascular biology with work centered on the temporal contribution of endothelial progenitor cells to the tumor vasculature. He's worked with Dr. Raffi, who is originally going to be the presenter, but we're very fortunate to get Dr. Nolan here, who worked very closely with Dr. Raffi at the Weill Cornell Medical College. His own work focuses on the distinctions between endothelial cells of various tissues, and he's a very accomplished flow cytometrist and a microscopist. If you could go to the next slide. Some of the questions which we can consider at the end of during the panel discussion is just the whole pivotal issue of cancer stem cells. To what extent is this valid, and for which types of tumors? Is something across all tumors, only some specific types? The other questions that could be considered for the panel discussion is what types of model systems should be used to investigate the mechanism of action of these different types of cells. What's the importance of cell type and context specificity for these different types of cells? And also a very pivotal question is what's the advantage of attacking the cancer stem cell over other conventional anti-tumor approaches? Other important issues are there predictors for effectiveness? As we talk a lot about how to accelerate our movement into the clinic, are there things that we can be doing with diagnostic markers, predictive biomarkers that will help us reach patients more quickly? And also, what kind of biomarkers or clinical endpoints should be used in the clinical trials testing these types of agents? So these are some food for thought that we might want to ask the distinguished panelists. In addition, I'm sure you're going to have many more questions as well. So without further ado, let me introduce Dr. Catriona Jamieson.
Thank you very much, Dr. Feigal, and also to the audience for being here and discussing this topic that's rather near and dear to my heart. I raced down here from my clinic at the Moores Cancer Center where I see patients, which keeps me on track. It's the major reality check. They happen to be rather well informed because we live in the hub of biotech. So people know more about their disease than you could ever imagine. They become experts on their disease and they teach us about it. So the idea of cancer stem cells was first suggested or posited in a reasonable way by John Dick in Toronto, and basically what he suggested was that there was a smaller population of cells within leukemia that could propagate disease in serial transplant models in mice. So that regenerative capacity of leukemia is something that we generally don't target using the anti-proliferative agents that make up our chemotherapeutic strategies, nor are they targeted with radiation therapy. So the reason is these cells that like to regenerate themselves, or in other words self-renew, tend to have the capacity to become dormant in protective niches. So they go to sleep. They're not dividing. They're like that house guest that never wants to leave. You have to give them a big prod. Okay, time to go. But they won't wake up. So that's the problem with cancer stem cells. And now they've been discovered in a number of tumors. A lot of controversy has existed about whether or not they're rare. I don't think the idea of a cancer stem cell that's capable of regenerating itself is predicated on being rare. I think the fact that they can self-renew, have enhanced survival capacity, can differentiate along different lineages and become dormant is what defines a cancer stem cell. So the great thing about being in the state is that we're allowed to be hopeful, not just for our patients, but for science, thanks in large part to the California Institute for Regenerative Medicine and the tireless efforts of Ellen and others who have really worked hard to change the paradigm for medicine and for science so that we're able to look at things from a stem cell angle. This has been made possible through training grants. So the most important thing is that we can fund talent that actually does the work, but also these buildings are kind of nice. So this is the building that we're working in. This is the Sanford Consortium for Regenerative Medicine, and it puts people together that have disparate abilities and ideas. Now, I think of cancer stem cells in an economic kind of way, because if you look at how we do single-agent therapy, when we look at molecularly targeted inhibitors like tyrosine kinase inhibitors, the minimum cost per year is about $100,000. Patients can pay up to $2,900 as their copay monthly. That's pretty difficult for most people to afford. So that $100,000 a year price tag is probably something that our economy won't be able to afford for much longer. But the thing with these targeted inhibitors, when given alone, is they generally don't target the cancer stem cell because they're only targeting one property and not the quiescent property. So these bad seeds, those dormant seeds, are the ones that need to be targeted. Those are the cancer stem cells, as alluded to in Nature this year. So these bad seeds like to hide out in bad soils, and that's what we call the niche. And there are certain elements in this malignant microenvironment that promote survival, self-renewal, and dormancy of these cells. And it looks like the cancer stem cells can remodel that niche, that can reside both in the osteoblastic area but also in the vascular niche, as Shaheen Rafi and Dr. Nolan and others have shown. So basically there are a number of pathways that are co-opted. One of them is the JAK2 pathway. We showed this was activated at the level of a hematopoietic or blood-forming stem cell in 2006. And then CIRM was interested in funding fellows to actually work on stem cell biology. So we had a very good fellow in the lab that worked on this project. And basically what we found was that this mutated JAK2 led to a disease when expressed at the level of the stem cell that recapitulated polycythemia vera. We were able to show that in a mouse model. If we took human stem cells and put that JAK2 gene in when it was mutated, we could get polycythemia vera. If we used a selective JAK2 inhibitor, we could knock down polycythemia vera expansion in those mice, and we could knock down the JAK2 mutation. Together with work done at Targogen, which is the local company that we collaborated with, and Gary Gilliland at Harvard, we were able to take that to phase 1 all the way up to phase 3 clinical trials. Now, the issue is long-term dosing with these inhibitors can be tough for people. And I think what we really need to do from the get-go is think of a combination strategy that will target these cells from different angles. We know that the JAK2 burden is important. We can reduce that in clinical trials. But because of long-term side effects, I think we need to look at combination strategies that will be shorter strategies. And that's not something that most drug companies want to think about. They want to have patients on their drug forever. So if you come up and say, no, actually, we want a combination strategy that will hit the cancer stem cells so hard that people will be able to come off drugs, that's not usually compatible with their bottom line. That's why CIRM is so important, because it helps us to devise strategies that potentially eradicate these cells that we can't develop otherwise. So we've had some success in reducing the fibrosis in the marrow, which is the scarring there. And this is not just the case for the drug that we were involved with, but also other JAK2 inhibitors. So I think we're making some strides, and we're about halfway there because we still have to prevent progression to what's called acute myeloid leukemia. And that's where reversion to an embryonic transcriptional program comes in. So what we see with cancer stem cells is they tend to think they're embryonic. They recapitulate some of the programs that you usually only see in the embryo. And an example of that kind of pathway is the hedgehog pathway. So the hedgehog pathway was described by Christian Nusslein-Volhard and others many years ago, and she won the Nobel Prize for pattern formation in the Drosophila, fruit fly in other words. And so she showed it was very important, and then other people said, well, we think it may be important in mouse hematopoietic stem cell maintenance. Tanisha Reya, who was recruited to San Diego from Duke and was previously at Harvard or at Stanford as well, showed in a mouse model of a leukemia called chronic myeloid leukemia that sonic hedgehog activation was absolutely vital for maintenance of the leukemia stem cells in that model. So basically what she showed was that if you inhibit that maintenance using a drug called cyclopamine, that you could really knock down the leukemia stem cell burden. So we wondered what happens in patients. Is this an independent arbiter of disease? Does it tell us if people are going to progress? And indeed, in blast crisis CML, which was previously thought to be untreatable, we found upregulation of the pathway, and specifically GLE2, which is a downstream transcription factor. And then our colleague John Dick, who, as you probably remember I mentioned, discovered leukemia stem cells, showed us that GLE2 is also overexpressed in leukemia stem cells in acute myeloid leukemia. And that was an independent prognostic risk factor. The non-leukemia stem cell population didn't express much GLE2, and then the hematopoietic stem cells, the normal ones, actually were also devoid of it. So you can see that there. We used GLE2 as a biomarker of response in xenograft models where we transplanted the leukemia stem cells, and that took us to a clinical trial. So this was the second target that derived from CIRM-funded research where we partnered with a local company. In this case, it was Pfizer, working with their specific hedgehog antagonist. In the clinical trial, what was very interesting is there are a lot of hopeful 69 and 70-year-olds who are very fit and they're willing to take a pill for their leukemia. And so the majority of patients that we enrolled on the hedgehog inhibitor clinical trial were patients with acute myeloid leukemia. We knew going into the trial that those with a higher GLE2 expression have a worse outcome. And so that was sufficient to get us going. You can see that the blast counts reduced by 50% in about 40% of patients on the trial. So that was enough to go into phase 2 trials, which is what we're doing now for high-risk MDS and AML. So these are pills. This is a pill that's now an option for people that are unfit, as we call it, and have AML. So they have other risk factors, and they're pretty happy to have a choice. The other thing that came from our HALT Leukemia grant, which is a disease team grant that Dennis Carson is the PI for and I'm the co-PI for, and we share with John Dick in Canada, is to target another embryonic gene called ROR1. And this Wnt5a receptor allows us to target the genes very selectively, or the cancer stem cells, because only cancer stem cells upregulate this gene. It's not expressed by normal adult tissues. So we were able to get almost all the way through the pipeline of development of a humanized monoclonal antibody targeting ROR1. We plan to file the IND early next year. The protocol for the clinical trial in CLL is already at our IRB. Now, this was based on a discovery made by Tom Kipps in chronic lymphocytic leukemia, where he found 94% of patients had actually upregulated this antigen, and it wasn't any epitope of that antigen. It was one specific epitope that happened to denote a cancer stem cell population. So we were able to go forward with guidance from CIRM to develop this humanized monoclonal antibody, and then are just getting a better handle on the PK in subsequent studies together with a local company. So. The interesting thing in the final slide was that this ROR1 antigen is upregulated in a number of malignancies in the cancer stem cell population in particular, as those malignancies, be it breast cancer, pancreatic cancer, decide to become invasive and metastasize. So we think that this is a very important way to work on cancer from this cancer stem cell angle, and it gets people from disparate areas working together. Actually, the final part is survival of the cancer stem cells. So again, with CIRM funding, this is early translational 2. We're developing a pan-BCL2 inhibitor that will target these cells. And the idea is that these cells are completely reliant on BCL2 for signaling. So if we knock down BCL2, we reduce the leukemic stem cells in the marrow and the spleen. With Sanford-Burnham investigator Maurizio Pellecchia, we've been able to purify this molecule and are scaling it up to GMP levels. So that, we think, is going to be an important base for doing clinical trials. So in terms of the pipeline of discovery, CIRM has made a tremendous difference for us at the Moores Cancer Center. Highlighted in red are the different biologics and small molecules that we've been developing as a result of this funding. So we're pretty excited about this and know that we're just getting started. So if you can just play the video, this will show you an example of a patient that was on the hedgehog trial.
Within 2 years, doctors told her that she was dying. Today, this grandmother is thriving. The chance meeting in San Diego that's saving lives.
Live from the Scripps Studios, this is San Diego's news source, 10 News.
And doctors say that she had about 2 months to live, but that was more than 2 years ago. A local woman now is alive and doing well. New at 6 o'clock, 10 News reporter Michael Chen looks at the local partnership that she credits with saving her life.
This is a symptom.
This bruising is one reminder of a disease that turned Linda Lowe's life upside down.
I'm bald and bruised, but I'm still fighting.
More than 2 years ago, the bone marrow disease living in her body, MDS, the same one affecting Good Morning America's Robin Roberts,
I literally fell down and couldn't get up.
turned into leukemia. Doctors told her she had a short time to live, likely months. Her condition was too grave for a bone marrow transplant. Her search for answers led to the UCSD Moores Cancer Center and a clinical trial born from a unique annual symposium mixing academia and the pharmaceutical industry. Because of the bonds formed, researchers called up Pfizer to compare notes on a new drug aimed at other cancers.
Together, we were able to reach a conclusion very quickly that this drug may be very effective in leukemia.
Just a year later, fast for the research world, Lowe was on an experimental drug that blocked cancer cells from cloning themselves. A year later, she qualified for a bone marrow transplant. She is now in remission.
I don't know that that would have happened if this collaboration had not taken place.
10 News has confirmed that an unprecedented project is moving forward. Right next to the Cancer Center, rising from this parking lot, will be an incubator of sorts. Big pharmaceutical companies and startups will be housed in the same building as academic.
So I didn't want to go on a long diatribe about UCSD, but the idea is that we can make a difference even if it's one person at a time, and that Linda is now essentially cured of her disease. She doesn't have any leukemia in her bone marrow. She's been through a lot, so she has a little bit of post-traumatic stress disorder, but I think she and her husband and her 3 grandchildren are quite grateful. So thank you for your attention. I'll be happy to chat during the panel.
Catriona, thank you very much. That was excellent and very inspiring. Our next speaker is going to be Dr. Robert Wechsler-Reya, and come on to the podium.
Okay. Great. So this will be a little bit of a change of pace, but hopefully on the same theme. I don't know how many of you were part of this group, but a few days ago, about 12 million Americans watched the mid-season finale of The Walking Dead. This is a picture from that TV series. And you may think that 12 million people, that's about 4% of the US population, watching a zombie show on TV is a little odd, perhaps unnecessary. But I'm here to tell you that actually we should all learn a little bit more about zombies. In fact, the Centers for Disease Control on their website has a section called Emergency Preparedness and Response, and if you look there, they have instructions for how to cope with the upcoming zombie apocalypse. They have a detailed pamphlet that describes what you should do in preparation. And among the other things that I've learned from reading this website is how to effectively kill a zombie. Now, if you know anything about zombies, the important thing to realize is that if you simply stab them in the heart or cut off their legs or hit them in some way, this will not be effective because zombies have the incredible tendency to just come back to life. The only way to really kill a zombie is to shoot it in the brain. Hope nobody had lunch recently. And so this is demonstrated here. And you can see that if you destroy the brain of a zombie, it will not come back. And the reason this is important is because this is the same strategy that I think we need to take in combating cancer. Because traditional cancer therapy is a lot like the way the extras in the movie try to kill the zombie. That is, you attack the bulk of the tumor with radiation and chemotherapy, and the thing simply grows back. In order to actually prevent this, we have to shoot at the brain of the cancer, the so-called cancer stem cell. If we kill the bulk of the tumor and we also kill the cell that propagates the tumor, we have a hope that the tumor won't actually grow back. And this notion of cancer stem cells, which Catriona introduced beautifully, has been around for a number of years. It's been rather controversial. And the reason it's been controversial is because science and medicine and cancer in particular is never as simple as we think it is. Different cancers behave differently, different cells within those cancers behave differently, and the generalizations always tend to be wrong. But the specifics may be right. There may be lessons to learn. And so the notion of cancer stem cells, originally discovered in leukemia, has now been demonstrated in many other cancers, both solid and liquid tumors. And we have been focusing on it for a number of years in the context of the tumor that we study, a pediatric brain tumor called Medulloblastoma. Medulloblastoma is the most common malignant brain tumor in kids. It's a very aggressive tumor that, if untreated, will grow rapidly, spread through the central nervous system, and kill the patient. The good news, of course, is that most patients with this disease are treated. In fact, they're treated extremely aggressively with a combination of surgery, radiation from the top of the head to the base of the spine, and high dose, usually a triple or quadruple cocktail of chemotherapies. This has resulted in very high cure rates, at least kids who survive for 5 years or more. The problem is a bunch of those kids die after 5 years, and the ones who survive have very severe long-term side effects, including cognitive deficits, endocrine disorders, and many of them actually go on to develop secondary cancers in other tissues later in life. And so although we can do some work here with a sledgehammer, what we need is a scalpel. We need a fine instrument to be able to kill the cancer stem cells, and prevent the disease from occurring without destroying the patient in the process. And so we have been using, among other models, a transgenic animal model called the patch mutant mouse, which was developed in the lab where I did my postdoc several years ago in Matt Scott's lab at Stanford. Now, patched is an element of the sonic hedgehog pathway that Catriona introduced a few minutes ago. Mutations in patched and in other elements of this pathway result in activation of the pathway. And since this pathway is involved in cell growth in many tissues, mutations in this pathway can lead to cancer. And so in the context of medulloblastoma, about a quarter of patients who have this disease have mutations in patched or in some other element of the hedgehog pathway. And the patched mutant mouse is a model for that type of disease. So animals that have mutations in patched go on to develop medulloblastoma. These animals have been very valuable for studying many aspects of the disease. They've been used to study the cells from which the tumors arise, looking at the early stages of tumorigenesis, and they were the primary platform for the testing of hedgehog antagonists that are now in clinical trials for this disease in kids. That work was done in Tom Curran's lab then at St. Jude, but now clinical trials are underway all over the world to study the role of hedgehog antagonists as therapy. And so the problem with those antagonists is that although many patients respond, almost all patients then have recurrence of their tumor. And so we still have a lot of work to do to treat these diseases. And we asked the question, could we do better by finding ways to target the cancer stem cells? And so the notion of cancer stem cells in brain tumors is almost as old as in leukemia. Peter Dirks at the University of Toronto a number of years ago demonstrated that cells in human brain tumors, a subset of the cells, express stem cell markers, including the antigen CD133, that these cells can undergo self-renewal in culture, that they can give rise to both neurons and glial cells, so they're multipotent, and most importantly, that if you sort those cells and transplant them into another animal, they will give rise to the tumors, whereas the CD133 lacking cells will not. Jeremy Rich and colleagues now at the Cleveland Clinic have shown that these cancer stem cells are resistant to radiation and chemotherapy. So together, all of this suggested that cancer stem cells might be a good thing to target in the context of treatment of medulloblastoma. And so we established a number of years ago an assay to try to identify cancer stem cells in our mouse model. And the way we did this is shown here. Simply take a tumor, from a patched mutant mouse, transplant it into the brain of another mouse, and ask if it gives rise to a tumor. And if you take enough tumor cells and transplant them, they will give rise to a secondary tumor that looks very much like the tumor that you started with. Now, this assay allows you to fractionate the tumor into different cell types and ask which of them actually propagates the tumor, which is the cancer stem cell. And we did this for many, many months using various markers and eventually came upon a marker that could help us enrich for the cancer stem cells. It's an antigen called CD15. And what we demonstrated is that we could isolate CD15 expressing cells. These are the cells shown in red here on this flow cytometry plot. And when we transplanted those cells, they always gave rise to tumors, whereas if we transplanted the CD15 lacking cells from the same animals, they never gave rise to tumors. So this suggested that the ability to form a tumor was resident in the CD15 population. Excuse me. Unlike the cancer stem cells that have been described in some other tumors, CD15 expressing cells are not quiescent. They are, in fact, they have an increased number of cells in the cell cycle, and that's just illustrated here. This is the percentage of CD15 positive and CD15 negative cells that are in the late phases of the cell cycle. They're also not stem-like in the sense that they cannot give rise to multiple cell types, neurons and glia. They only give rise to neurons. However, these are genuinely the cells that propagate the tumor, and so from a clinical standpoint, these are the cells that you'd want to be able to target. We've gone on to demonstrate that a similar population of cells is present in human medulloblastomas of various histological subtypes, and through bioinformatic analysis, we analyzed the genes that are expressed in CD15 positive cells and found that those genes predict a poor outcome in patients with medulloblastoma, and that's illustrated here. So patients who have more of the genes that are found in CD15 positive cells, those patients tend to do worse. This suggests that cancer stem cells of this type may be driving the growth of the tumor. So we published this work a number of years ago, and the summary of that part of the talk is that hedgehog-associated tumors can be propagated by cancer stem cells, that these cells express CD15. They're not rare, they're not quiescent, they're not stem-like, but they are the cells that propagate the tumor. And if we can target these cells, our hope is that we might be able to make therapies more effective. And so the question is now, how do you do that? And we've tried a number of strategies to be able to target CD15 positive cells in a tumor growing in an animal. We've conjugated toxins to the CD15 antibody to see if that would work, and that has not been effective. What we've done instead is to begin to study the properties of these cells and try to figure out, is there something special about them that we might be able to use to eradicate them? And so in order to determine what are the unique properties of cancer stem cells, we took a bunch of cancer stem cells from mouse tumors. We compared them to the non-cancer stem cells from the same tumor. And what we found is that there's a set of genes that has elevated expression in the cancer stem cells compared to the non-cancer stem cells. And these included a whole variety of genes, many of which were associated with the cell cycle. And some of those are illustrated on the bottom here. And the ones that we focused on are the ones for which there are good drugs, good inhibitors. And those are the aurora and polo-like kinases. Now, aurora kinases and polo-like kinases play critical roles in cell division. They also play important roles in asymmetric division that's important for self-renewal. And so we thought these would be really good, a really good focus for our attempt to eradicate cancer stem cells. And I won't go through the details, but each of these genes, aurora kinase A, aurora kinase B, and polo-like kinase 1, have overlapping but distinct functions in the context of the late phases of the cell cycle when cells are beginning to divide. The important thing is that we could demonstrate that aurora kinases and polo-like kinase 1 had elevated expression in cancer stem cells compared to non-cancer stem cells from every tumor we looked at, and that there were drugs that were available that we could use to test targeting of these enzymes. And so the drugs are shown here. These are a subset of the Aurora and polo-like kinase antagonists that are now in clinical trials, and they're in clinical trials for a wide variety of types of cancer, including prostate, leukemia, lung, and colorectal cancer. And so we began to ask the question, can these drugs be used to eliminate the cancer stem cell and reduce the growth of the tumor? And what I'm showing you here is that in an in vitro assay of proliferation, we can dramatically inhibit the growth of the cells from our tumors using either the Aurora or the polo-like kinase antagonist. These antagonists are effective at killing the tumor cells, but they do nothing to normal neurons, which are some of the other cells that they're going to encounter when they're in the brain. And that's shown here, whereas you can see an increase in cell death with the tumor cells, normal neurons don't really care. These antagonists are likely to be effective, but not on their own, and this has been shown in many different contexts. And so our next goal was to see if we could combine them with other therapies and see if we could improve the efficacy through combination approaches. And so this is one of the hedgehog antagonists that's in clinical trials. And we showed that we could get inhibition of tumor growth with the hedgehog antagonist alone, but we could get much better inhibition when we combined it with, in this case, the Aurora, and we see similar effects with the polo-like kinase inhibitors. And so this allows us to either have more effective kill of the tumor cells or to reduce the dose of the antagonist, of the hedgehog antagonist, so we reduce some of the side effects. And that second strategy is particularly important when we try to combine it with conventional chemo and radiation therapy, because those are the toxic therapies that are used now that are clearly effective but have devastating side effects. And so we tested the effects of combining our antagonists with radiation, and shown here is the fact that we can get a significant kill of tumor cells with radiation alone, but we can enhance that kill when we combine it with Aurora kinase or polo-like kinase antagonists. And the same thing is true for chemotherapy. This is one of the cocktail of drugs that's used to treat medulloblastoma, and you can see that on its own it kills tumor cells, but in combination with the, in this case, polo-like kinase antagonist, we get a dramatic increase in the efficacy. In fact, shown on the bottom, these IC50 numbers are the efficacy of the drug, and we increase it about 50-fold by combining these two agents. Importantly, we can get kill of not just mouse medulloblastoma cells, but through a collaboration with physicians at Rady Children's Hospital, we've been able to establish xenografts of patient cells. So we go to the operating room, we collect the cells, we bring them back to the lab, and we implant them into a brain of a mouse, and then we passage them from mouse to mouse. These cells maintain many of the properties of the original patient's tumor, and they're an incredibly valuable tool for studying the efficacy of drugs. And what you can see here is that in the context of one of these, a hedgehog-driven tumor, we can see significant inhibition of growth, not just by the hedgehog antagonist, but also by the polo-like and Aurora kinase antagonists. And in fact, we've established one of these from a patient that is resistant to hedgehog antagonists and still responds to the Aurora and polo antagonists. And finally, we've tested the efficacy of these drugs in an animal, in tumors growing in an animal. And what you can see here is that animals that are treated with vehicle, the tumors grow dramatically. When animals are treated with the polo kinase antagonist, we see much less growth of the tumor. When we excise the tumors at the end of the experiment, the ones in the treated animals are much smaller, and that's quantified here. You can see that there's a dramatic inhibition of growth just with this agent alone. And as I mentioned, I think combination therapy is going to be the key to eradicating this disease. But what we can show is that even with a single agent, we're getting shrinkage of the tumor in vivo. And that, to me, is the first step in beginning to develop a combination therapy that will be effective for patients. So just to summarize what I've told you, medulloblastoma tumor-initiating cells or cancer stem cells have high levels of Aurora and polo-like kinases. Antagonists of these kinases inhibit growth of tumor cells, both in vitro and in vivo. They block growth of human hedgehog-dependent tumors, and they represent an important new avenue for therapy for medulloblastoma. So we're continuing our preclinical studies and also beginning to talk to both physicians and drug companies about getting these into clinical trials for patients. So just to summarize, just to conclude, I want to acknowledge the people who have done this work. The identification of cancer stem cells in medulloblastoma mouse models was done by Tracy Ann Reed, a former postdoc of mine, who's now faculty at Emory. And then the characterization of the effects of Aurora and polo-like kinase antagonists was the work of Shirley Markant, an incredibly talented graduate student who's now doing a postdoc at Pfizer. This is a picture of my lab on the beach, for a change. And you can see both our collaborators shown on the right here and our funding sources. And I want to acknowledge particularly CIRM, which allowed me to move my lab from Duke to here and has provided an incredible amount of funding and support for us to do the studies that we're doing. So thanks very much.
We're holding questions till the very end just because, as you can tell, we're running a little bit short on time. So let me go ahead and introduce, and I will remember the anti-zombie approach. That's very innovative. Our last speaker before we go into the panel discussion is Dr. Daniel Nolan.
Good afternoon, everyone. Shaheen sends his regrets. He was held back in New York at the last minute. One of the major donors to the university wanted to hold a meeting, and when you have a donor who has donated that much money, you respect every request that he asks of you. So to get into where we are going with the endothelial cells and how they relate to cancer, we're going to take a step back for a minute and look at what lessons can be learned from what we in the field have been finding out about endothelial cells and stem cells in general. Healthy stem cells, not the cancer stem cells yet, but we'll see what we're learning here and how they can apply. Just got to figure out what button to push. So in the last 10 to 12 years, the vascular biology field has been coming up with more and more examples of developmental paradigms where specific tissues are reliant on the growth factors derived from the endothelial cells for their proper tissue development. So the endothelial cells in a developmental paradigm will signal to the tissues when they are prepared to support their growth. And this has been shown in the liver as far back as Matsumoto's paper in 2001, and also as recently as the Testes paper written by Kuhl. And there are also several other examples in the literature, including heart and intestine, et cetera. So the question now is, why should the endothelial cells signal tissue regeneration? And the answer is pretty logical. If you have a tissue growth that grows without blood vessel support, it is going to get too large, it's going to become necrotic and die. But if you have a coordinated crosstalk from the tissue back with the blood vessel and the tissue will now grow in unison, in synchrony. And this synchrony is governed by growth factors. This is going on its own, I'm sorry. This synchronicity is governed by growth factors from the tissue itself going to the blood vessel, and the blood vessel then replying with additional growth factors on its own. And these growth factors that are being derived from the blood vessels, we coined a term several years ago called angiocrine factors. It's to specify the growth factors that are being secreted directly from the endothelial cells. And that it is this maintenance of tissue in both normal and healthy adults that's maintained in the adult. In a developmental context, evolution, when it figures something out, it likes to reuse it constantly. So this is something that's been found in development, and when you get to an adult model, we're going to show you now that it still applies. That a tissue regeneration model, when you have a wound, has the same reliance on the angiocrine factors coming from the blood vessels for tissue regeneration. So one of the things that we set out to understand was how complex is this cadre of signaling molecules, these angiocrine factors coming from the blood vessels. Was it a handful of factors, or was it a large number of factors? So one of the works that we set upon doing was to understand the complexity of the angiocrine signaling coming from the endothelial cells. So what we endeavored to do was to profile the endothelial cells from an in vivo source. So we were able to isolate the endothelial cells. These have never been cultured. They have never been manipulated other than simply isolating them to a high degree of purity from the animal. And what we found was a tremendous amount of heterogeneity from tissue to tissue. So some of it's cut off down here, but you can see that there are 9 different tissues that we looked at, and there was a profound difference between the different tissues. And one thing to note is that this is only a selective list of all the factors. We simply couldn't put all of them up here. There were approximately 260 divergent signals coming from the 9 different tissues that we looked at. Now, one of the things that's very important to consider when you're looking at the factors that are secreted by the endothelial cells to the surrounding tissues is that there are no unique factors that we were able to find. It was one of the first things that everyone asked us. What's brain specific? What's liver specific? What's lung specific? And my answer is nothing. There is no single factor that is unique to a given tissue, and that the uniqueness of the vascular niche is governed by the combinatorial effect of hundreds of factors being supplied to the surrounding milieu. And the way to think of it is more of the teeth of a key as opposed to single unique factors that are governing the vascular stem cell niche. And these patterns are also similar when you look at other gene clusters. If you look at the metabolic profile of the cells, the surface receptors, and their various forms of regulation. All of the endothelial cell beds in each different tissue are quite unique. So the next question that we wanted to ask was, is this a standard set of cytokines and growth factors and matrix proteins and remodelers that all of the endothelial cells in these tissues are providing, or are they dynamic? Do they change? And the answer was very overwhelming in the sense that they are very dynamic. So we chose 2 models that we are very well experienced in dealing with. Okay, it did come up on that screen. So we're looking at the radiation-induced myeloablation in this first column. So in the this is bone marrow at steady state, 10 days after radiation, and 28 days after radiation as it's recovering from that myeloablative stress. Now remember, these are primary cells isolated directly out of the animal. And what we're finding when we compare the bone marrow to the liver after partial hepatectomy, 2 days after regeneration and 6 days after it has begun regeneration, you can see that there is a profound change in the angiocrine profile of the vasculature in how it is supporting the regeneration and remodeling of that tissue. It is unique at day 10, it is unique at day 28. Not only do you have specific genes that are upregulated at early time points, around day 10 you have somewhere near the peak of regeneration, and when you look at day 10 as the regeneration starts to taper off, you see that there are also a unique signature. In contrast, these groups of genes that came up in the bone marrow were unique to the bone marrow and not to the liver. And when you look at this at a genome-wide level, there is less than a 15% overlap in upregulated genes between these 2 tissue types. So the angiocrine factors are very dynamic, and they are responsible for both the initiation of tissue regeneration and also the resolution phase as the tissue comes back to its normal size and function. So what we were able to do with this is we wanted to bring it in vitro. We wanted to take this phenomenon that we can see in the body and in vivo and bring it to a point where we can study it in vitro, because that would make a very manipulatable model for stem cell biology. And this was a fortuitous discovery by Shaheen several years ago by accidentally leaving a vial of plate of cells in the incubator, and it turns out that this particular control well, what did I just do? There we go. Had E4-ORF1 specifically. Now E4-ORF1 is being studied. It's an adenoviral protein in various different cell types, and in each cell type that scientists are looking at it, it has quite unique features. In the specific aspect of endothelial cells, where the endothelial cells themselves have quite a unique metabolism compared to other cell types, it gives a very, very modest activation of AKT. And for those of you not familiar with the biology of the endothelial cell, the AKT pathway is a main signaling hub for all things endothelial, namely VEGF-A, FGF2. There is a very minimal and a very specific activation of the AKT pathway and not other pathways. And this AKT activation is sufficient to confer a serum and cytokine-free survival effect on endothelial cells. And we have now termed these endothelial cells the Vera-Vec series of endothelial cells. And where this comes into play with stem cells is that we can now take a monolayer of these Vera-Vec endothelial cells in serum-free conditions after we bring them to a nice confluent monolayer, plate stem cells on them, and these stem cells will proliferate quite well. And the biology of these expanded stem cells is quite profound. And this was work done by Dr. Jason Butler, a colleague of mine, also coming out of Shaheen's lab. And what you're looking at here is a confluent monolayer of the endothelial cells plated with hematopoietic stem cells on top of them. And after 28 days, I believe, if my numbers are correct, this is 1/10,000th of the actual expanded population. If you look at the expansion numbers here in comparison to cytokine controls, you can see that the expansion on the Vera-Vec endothelial cells are far greater than cytokine-mediated alone. And when Jason went and transplanted these cells, this is where he gets some of his best results. Now, what I'd like to draw your attention to in what he's shown here is that when you have cytokine-mediated expanded hematopoietic stem cells and you do a transplant into recipient mice, what you see here in the black and the gray bars are short and mid-term engraftment at 4 and 8 weeks. And you'll see also that there are the white bars at 48 weeks. This will be a long-term engraftment. And when you have cytokine-mediated expansion, you see no long-term engraftment. When you compare this to the endothelial cell-mediated expansion, you see quite a robust contribution to long-term engraftment. But what's even more exciting about that is when you compare it back to the same number of primary isolated stem cells, you actually have, in this case, 2.5-fold more long-term engrafting stem cells in the expanded population compared to your starting material. The cells that expand on the endothelial cells are actually selected for the most primitive population. So after this paper was published, Jason started to work on doing this in a human context. And he started off with umbilical cord blood. I'm actually proud to say this is my son's cord blood you're looking at here. He was born down the hall, and his cord blood was hand-delivered for this experiment. And you can see that the endothelial cells, the E4F1 endothelial cells, were capable of expanding Nathaniel's cord blood far greater than cytokines alone. And again, as we saw with the mouse, you can see that they are CD34 high, CD49F positive, 45RA negative, and they also give rise to a multi-lineage commitment. One of the things that is very pertinent when it comes to expansion of the hematopoietic cells, many protocols will cause the myeloid preference in the expanded cells. However, these cells give rise to a multi-lineage commitment, maintaining their pluripotency. And importantly, again, he saw that when you quantitate the number of long-term repopulating stem cells in the expanded field, that you had approximately an 8-fold increase in the long-term repopulating stem cells in the expanded cells. So we're looking at the vascular stem cell niche and how it relates to stem cells. And we have various other examples of how this works in the liver, how it works in the lung, et cetera. And we want to know what the ramifications are of the stem cell vascular niche on cancer. And several days ago, we just got word that a paper has been accepted in Cancer Cell by the title of Angiocrine Factors Deployed by Tumor Vascular Niche Induced B-cell Lymphoma Invasiveness and Chemoresistance. And the authors on this are Zhongwei Cao and Bi-Sen Ding in Shaheen's lab. And this is going to be one of the first times this data is presented, and it should be in press in a few months in Cancer Cell. So the question that Bi-Sen and Zhongwei were trying to answer was, beyond the genetics, beyond the mutations, what dictates the aggressiveness of tumor cells, specifically in the context of our favorite stromal component, the endothelial cell. And one of the things that they did was they started looking at how xenograft cells are cultured in the lab. And they were looking at the contributions, and actually the deleterious contributions, of serum in the media that is used to grow these various cancer cells. And the first thing that they found was that, as they have labeled here in their figure, the lymphoma cells grown on endothelial cells grew far better in serum-free conditions compared to standard culture conditions of lymphoma cells in these medias. And that these cells that are grown on the endothelial cells in serum-free conditions are able to behave more like a cancer stem cell in methylcellulose assays compared to cells grown in serum. And this is actually a video of what these cells look like. So these are the leukemic cells in red. These are human leukemic cells grown on the Vera-Vec endothelial cells. And this is in complete serum-free conditions. In the adjacent well, there were leukemic cells that after 24, 48 hours were dead because they could not be sustained in media without the serum. And in this case, the cells grew far faster by the angiocrine factor supplied by the endothelium. So when these cells were taken by Zhongwei and Bisen and implanted into the animals, they started off with the standard controls of injecting the cells grown in basic serum-free conditions. And you can see how the mortality of the animals were increased when co-cultured in a co-culture model with the endothelial cells. And you can see the profound enhancement in aggressiveness of these cells that were grown in this manner. So what they were looking for were the phenotypes of these more aggressive cells now. So the co-culture with the endothelial cells alters the lymphomas, and specifically in a jagged manner. So not to belabor the point too much, but these CD44, CSF1R, IGF1R lymphoma cells are considered a more less aggressive phenotype of cells. They are not very well enabled in their engraftment into animal models. And these cells were co-cultured with the endothelial cells, and they were able to revert back to a more aggressive phenotype. So what he was able to find was that the CSF1R and IGF1R phenotype was restored in 10% of the population. These cells were phenotypically more aggressive. And this specifically is done by a jagged one mediated phenomenon. Now this comes into important, I'm not going to go into the pathways of how this actually works, other than to say that jagged one is only going to be able to stimulate the receptors on the cancer cells if the cells are in direct contact. This is not a secreted factor. This is a cell membrane bound ligand. So the cells must be in direct contact for this signaling to take place. And if you eliminate jagged on the endothelial cells, these cells are no longer capable of regaining their more aggressive phenotype. And he went further to take this into single cell colonies, where he took a single cell that was the less aggressive phenotype, plated them on endothelial cells, and he was able to restore in a clonal fashion the aggressive triple positive cell type. And again, there's a tremendous amount of data in the paper looking into the signaling with jagged, and they were able to regain the aggressiveness in the context of growing them in a serum-free manner on the Vera-Vec endothelial cells. And the final piece of data that they go into is that the tumor microvasculature creates a chemoresistant niche. And again, they're doing this in the context of the jagged one, because the cells need to be in direct contact. And you can see in these survival curves here, they did it on both sides. And on one aspect, they deleted the FGFR1, which is responsible for stimulating the endothelial cells to present the jagged one. And they also took it from the context of deleting the jagged one specifically on the endothelial cells. And in both contexts, when you disrupt the interaction between the cancer stem cell and the endothelial cell, you now lose this chemoresistance. And that this direct contact that the vasculature, the vascular stem cell niche in the tumor, creates a safe haven for the tumor cells. So where we come to from this is that each vascular bed has a unique microvasculature, and specifically in the context of how it affects its microdomain, the vascular stem cell niche around it. This angiocrine profile is dynamic to the needs of the tissue and also the needs of the tumor, and that the Vera-Vec endothelial cells that Zhongwei and Bisen were using is capable of recapitulating the vascular stem cell niche, And they are also capable of enhancing cultures of numerous cancer types. This paper was focused on human leukemias and also a mouse lymphoma model. However, this is also applicable to various other models. And that the vasculature of tumors provides a chemoresistant niche for the cancer cells. And to sum it up all together, the real take-home message, if you had to sum it up in one statement, is that the analysis of tumor cells, both in vitro and in vivo, when done as a monoculture, is insufficient. And that more complex assays need to be done to be able to better recapitulate the biology of what the tumors are behaving as in vivo. And part of that aspect is the disruption of the vascular stem cell niche in tumors. And we also have a poster and a booth here, 32297 and 205, if you'd like to come and talk to us more directly about this. And I'd like to thank the people that worked on this on both the Cornell side and on the Angiocrine Bioscience side. And I assume that at this point we're moving on to a panel discussion.
Yes, thank you very much.
Thank you.
So those were excellent talks, so thank you very much. Why don't I open it up for questions right now? And I assume we have a microphone that can be circulated? Okay. Questions? There's a stand right in the middle of the aisle there. People want to ask questions. And we have, if you could just state your name too, that would be great.
Yes, so my name is Margarita Gutova from City of Hope Beckman Research Institute. And my question is to Wechsler-Reya. So you've nicely shown that medulloblastoma cell growth can be inhibited by kinase inhibitors, and also this effect can be shown nicely in vivo. However, I was wondering if you have a chance to look at the remaining tumors that comes after the therapy. So the tumors or in vitro cells that you target with your antagonists and small molecules, what is remained after treatments? And, you know, is this population CD15 positive? Or what kind of population is it? So what will be the second round of chemotherapy that we will propose to target recurrent population of cells? And if this population will be cancer cell-like or not?
So that was a very good question, and I think the in vitro studies we've done, we're able to completely eliminate tumor growth and, in fact, cell viability when we treat the cells in culture. So the Aurora and Polo-like kinase antagonists effectively kill the cancer stem cells, the CD15 positive cells, but they also kill the non-cancer stem cells. And that begs the question, how specific do we need to be? If we have a therapy that can kill both, to me that's better than a therapy that just kills the cancer stem cell. Now in vivo, I think the story may be more complicated. In the studies I showed you, we were also able to dramatically shrink the growth of the tumors. What we'd really like to do is be able to treat mice with intracranial tumors, because the key in treating brain tumors is to get a drug that actually accumulates in the brain. And so the antagonists that we used are not blood-brain barrier penetrant. Oh. And so asking whether these specific drugs are going to be effective therapies for the disease, I think the answer may be no. There are a number of other Aurora kinase and Polo-like kinase antagonists that are available. Some of them are in trials, and we're beginning to explore those to see if we can treat the intracranial tumors. So the question of whether there will be a resistant population remains to be seen. My feeling is that combination therapies are the way to circumvent populations that are resistant to a single agent. And practically speaking, in the clinic, we're unlikely to drop anytime soon the radiation and the chemotherapy that are currently being used. What we may be able to do is lower the doses so that the side effects are not as severe by combining them with other agents.
Okay, thank you. Also, I have a question to the third speaker, a short question. can you comment on angiogenesis independent tumors that don't have angiogenesis and they more stem cell like? They not vascularized, that they don't have endothelial vascular endothelial cells, but they are cancer-like, a cancer stem cell-like.
So one thing to keep in mind when you talk about angiogenesis is that there are two distinct types of angiogenesis. There is an inductive angiogenesis, and there is the proliferative angiogenesis. And on the side that when most people talk about angiogenesis, especially in the cancer field, they're talking about proliferative angiogenesis. But that is missing the entire other part of the instructive angiogenesis, where the endothelial cells are providing all of these different growth factors. So you're talking about tumors that co-opt vessels as opposed to forming the formation of new vessels. So it is still possible that in those contexts that they are stimulating the vasculature, which are constantly producing growth factors and co-opting those growth factors as well as their blood supply.
Okay, thank you.
I think there was, can you come to the microphone? Thanks.
Thank you. First, I want to compliment you on the evolution of the discussion. I think that we heard two interesting discussions about new strategies for cancer, and the third one was practically the one that started talking about stem cells and about the different aspects. I want to play for a minute the devil's advocate and say that the first two presentations were talking about strategies which are intuitive, which are the next step for anti-cancer development that has been done in the next at least 10 to 20 years. It's finding new targets. It's looking for how to stop this specific cancer activity. While it was less talking about their stemness, about the stem cell activities or features that these cells differ from the other cancer cells. The third one was more looking at normal stem cells, or how stem cells function can be used as a strategy to attack these stem cells. I would ask all three participants how they move from anti-cancer to anti-stem cell, or using function of cancer cells versus function of stem cells in order to develop new strategies for cancer.
Why don't we start with Dr. Jamieson?
Right. So we define stem cells often phenotypically, but I think your point is well taken that they have to be defined functionally, meaning do they clone themselves, or in other words, self-renew? Can they undergo differentiation along different lineages? Can they home to protective niches where they have the capacity to be quiescent even if they're not quiescent all the time? So, and do they have enhanced survival characteristics? So those are the four things that we look for to say that it's a cancer stem cell. So we have in vitro stromal co-culture assays, but most importantly in vivo assays to test the capacity of a normal human hematopoietic stem cell to engraft a mouse long term. So that involves using mouse models that have no T, B, or NK cells, and we transplant those cells into the mice at birth intrahepatically so that they're in a nice milieu that they like. And in that setting, they can make an adaptive human immune system and engraft for over 16 weeks in the animal and serially transplant. Cancer stem cells do that better because they don't have the Hayflick limit. So when you think of the telomere field, and what Elizabeth Blackburn won the Nobel Prize for was to say that normal stem cells can only divide about 5 times or undergo self-renewing divisions 5 times before they just cut off their capacity to protect the ends of the chromosomes through telomerase. So unlike those cells, cancer stem cells don't have a Hayflick limit. They keep self-renewing. So we do serial transplantation more than once. We have an in vitro replating assay as a measure of cancer stem cell activity, the ability of one colony to make another colony when we plate it, and then again and again. We can do that over 5 times. In serial transplantation, we do that in vivo. So all the work that I talked about in terms of cancer stem cell identification a priori required the functional validation of that population as a cancer stem cell in serial transplantation models and in limiting dilution assays. So we're very particular about what we call a cancer stem cell based on function. So all the cells that we sorted had the phenotype that we know can serially transplant disease, and we also tested those cells to show that they could serially transplant disease. In our treatment models, preclinically, we showed that we inhibited serial transplantation potential for the hedgehog antagonist when combined with a BCR-ABL antagonist called dasatinib. And that's why we felt that that was sufficient to go into clinical trials. What we didn't expect in the clinical trials was that it would work so well alone. We really expected that we would have to have a combination strategy, but in acute myeloid leukemia, they were so exquisitely sensitive to this hedgehog inhibitor that seemed to get the cells out of quiescence, out of the bone marrow niche, and into the peripheral blood where they underwent terminal differentiation, and that's why the patient I showed you did so well. So those are the mechanisms we're trying to understand now in the bench to bedside and back again kind of way, where we can take the patients that responded beautifully and say, why did they respond so well and the other ones not? So that science is circular. We always go back to the bench and look for functional validation. So I think your point's well taken.
Dr. Wechsler-Reya, do you have anything to add?
I'd just say that I think there's a tremendous amount to be learned when you are considering cancer stem cells. It's very important to understand the literature and the characteristics of normal stem cells. It's also important to understand the distinctions and not to assume that a cancer stem cell will behave like or be vulnerable to the same therapies as a normal stem cell. And so in each of the models that we study, in each of the both patient-derived xenograft models and animal models, we functionally define a cell as in terms of its ability to propagate the tumor before we call it a cancer stem cell. And then we try approaches that are designed to target that cell. Ideally, you'd want to be able to target the cancer stem cell and spare the normal stem cell. And so as a comparator, we always use normal progenitors or stem cells to make sure that there's a therapeutic window, that there's a differential kill of the cancer cells versus the normal cells. And so stem cell biology is incredibly instructive, but I think for each particular disease type, we need to understand the phenotype and the function of the cancer stem cells in that tumor.
Dr. Nolan, anything to add to that?
One small point, that the answer has been pretty well addressed to this, but one thing to keep in mind when you are trying to learn things from the stem cell field and bring it into the cancer stem cell field, there are dramatic differences between the populations. There are a great number of things that can be learned, but nothing can be taken for granted when you move into the cancer stem cell field. The stem cell biology field, a lot of these stem cell systems, they work like clockwork. When you're looking at red blood cell differentiation from the CD34 positive stem cell, there is a 5-day differentiation to a terminally differentiated functional cell, period. When you look at the same context in a cancer stem cell, all bets are off. There is a great opportunity to learn about overall mechanism, but you need to be able to go back and look at the biology of the cell, because if you look at what Bioscience and Zhongwei did in this publication they just got published, they took what, I don't want to get caught up on semantics, but potentially a differentiated cell. And this differentiated cell, by being basically coddled by the endothelial cells, were able to regain a more cancer stem cell-like feature. So that does not happen in stem cell biology. There is no reversion, naturally.
We're running a little bit over, but one last question.
My name is Ubaldo Soto from Loma Linda University. A short question. I missed here a general comment about metastasis. Do you think that in a new niche, the treatment probably that we are try for the original tumor will be not working, or that the stem cells marker or characteristic in the new niche could be different, and therefore we have an extra challenge, because actually metastasis is one of the big problems in treatment with cancer, more sometimes than the initial tumors.
Yeah, so I think that we have cancer stem cells that get stronger, basically, and they co-opt more stem cell properties and become more primitive as they decide to invade and metastasize. And that's where we see the upregulation of ROR1. ROR1 is really in these metastatic malignancies. So I think you're making an excellent point that the cancer stem cells in the primary niche aren't necessarily the same in terms of their functional characteristics as those in the metastatic niche. And so in the metastatic niche, the real question is, are they contributing to their own malignant microenvironment? And that's been an area of some controversy, but I think is very interesting. You know, can they help to basically corrupt that environment once they land in there, and what is it that's attracting them there? But I think nonetheless they've given themselves away by overexpression of genes like ROR1. We can target that, and there are other genes that are upregulated once they decide to invade and metastasize. So I think that that population needs to be studied separately, and in the field, unfortunately, they've been lumped together a little too often. So I think we have to consider them separately. There was a paper about circulating cancer stem cells by Andreas Trump showing that they upregulate the don't eat me signal called CD47. So they can circulate impervious to phagocytosis by macrophages, and they've upregulated other markers as well. So I think they can be detected in the blood, they can be detected at the metastatic site and targeted there, but we need to more specifically study those populations.
Any other comments?
I'll just sort of echo that. I think that cancer stem cells are a dynamic population. They don't stay the same throughout the course of tumorigenesis, either in the primary tumor or in the metastatic site. And so we're going to have to be equally aggressive and equally adaptive to try to kill them. It's very clear from the little profiling that has been done that metastatic lesions have a whole set of different dependencies, different mutations. And so to be able to effectively target metastatic disease, which as you said is critical, I think we're going to have to develop better models and better tools to be able to study metastases.
Okay, well, anyway, I want to once again thank the speakers for the excellent presentations and to you for the very good discussion. Thank you very much.